welcome to the second symposium on the second day. And I'm sure you are all still very eager to listen to all the speakers. And in this session, the topic is from dengue research to clinical implementation. And this is presented by the Indonesian Paediatric Society. And my name is Utarini, and I'm very honored to be able to chair this session. So thank you to the organizer. Let's see the three speakers background, and I will introduce the speakers. The first one, Dr. Tedjo Sasmono. He's a senior research fellow and also the dengue unit head of the Eijkman Institute in Indonesia. And then we'll have our second speaker, Dr. Ida Safitri, a pediatrician from Gajah Mada University. And she's also a peer reviewer of the WHO dengue guideline in 2009. And then our last speakers today in this session is Dr. Anggraini Alam. She's a pediatrician from Pajajaran University and head of infection and tropical disease of the IPS. I'm sure you are already very eager to listen to the session, so I will not hold for long. Because this session we have 1.5 hours long. So we hope that from each speaker would use about 20-25 minutes at most presentation. So that will leave us enough time for the participants to be able to pose questions and then have the answers from you. While we are listening to the speakers, of course, all the participants are also welcome to already start typing your questions using the Q&A. All right, so we'll start with the first speaker. Dr. Tedjo Sasmono will be presenting about distinct dengue epidemiology and viral characteristics in the Western, Central, and Eastern regions of Indonesia. Please, Dr. Tedjo Sasmono.
Okay, thank you for the opportunity, Prof. Adi Utarini. I will share my screen. First of all, I'd like to thank the organizer for giving me the opportunity to share our study. So I will be talking about the dengue epidemiology in Indonesia. So we have learned from previous speakers from Thailand, and then I would like to present my slide, my presentation here, mostly talking about the epidemiology of dengue in Indonesia. So I will start with the introduction. I think we all knows about the dengue already. So, but this is specifically for Indonesia. Indonesia reports the highest number of dengue annually in Asia. So we are the first in Asia, and while we ranked second in the world after Brazil, and also the global dengue risk map still classified Indonesia as a frequent or continuous risk for international travelers. So it is a health problem in Indonesia with year-round transmission. We live with dengue every day. And so far, the prevention depends on the factor controls and the community participation. And hopefully, we can have the vaccine to be a program of our government. And then this is for the clinician, the early diagnostic important for better patient management. And for the point of view of epidemiology, the virological surveillance can help predict the epidemics, but it's still not widely performed in Indonesia. And then this is an introduction about Indonesia. So we are an archipelagic country of a lot of islands, 18,000 islands. And then we also ethnically diverse. We have a lot of ethnic groups, and it's roughly the country can be divided into 3 regions: the Western, Central, and Eastern region of Indonesia. So I'm living in Jakarta, which is in here, in the western area of Indonesia. And we have a wide range of course, geographic, climatics, and demographic conditions in Indonesia because of this very vast archipelago. And of course, there are also inequalities in infrastructures and socioeconomic development. And the western part here tend to be more densely populated and more developed in terms of the infrastructure and public health, in contrast to the eastern counterparts here. So this inequality has been a problem in Indonesia, and overall, whether this diverse condition influence the epidemiological characteristic of dengue or other arboviruses in Indonesia is unknown. So dengue has been affecting people in Indonesia since 53 years ago. So it first detected in 1968 in Jakarta and Surabaya, and then increasing number of cases reported and then spread outside Java. And now dengue have been reported in all 34 provinces in Indonesia. So the incident rates drastically increased from 0.05 in 1968 until 86 in 2016 here, but the case fatality rate decreased over time. And we also have periodic outbreaks of dengue, typically between 8 or 6 years. Some people are saying it's 5 years cycles, but if we look at the data, it's kind of 6, 8 years. So to understand, to determine and compare the clinical, demographic, and virological features of dengue in different regions of Indonesia within the same period, we conducted the molecular epidemiology study, and the study is also to screen for possible arboviruses infection in Indonesia. So this study is a cross-sectional, and we conducted study in 3 cities in Indonesia, representing Western, Central, and Eastern Indonesia. So the first city is Batam in the Rio Island in province. Yeah, this one is the Batam in West, and then Banjarmasin in South Kalimantan is representing the Central Indonesia, and Ambon in Maluku representing the Eastern Indonesia. So this is the city of Batam, and Batam is a port city in the island of Batam. Excuse me. And the Banjarmasin is a city in Kalimantan. The city is divided, sorry, by a large river there, while Ambon is a coastal city as well in Ambon Island. Excuse me. So the study time, we conducted the study last few years in 2017 until 2019, and the inclusion criteria was the children and adults between the age 1 until 65 with a fever over 38 degree less than 5 days and symptoms suggestive of dengue disease. This is a quite broad inclusion criteria because we want also to get the patient with possible other arboviruses infection. And we exclude patient with the symptoms of upper respiratory or gastrointestinal tract infection and unwilling to participate to the study. So we exclude the flu and also the typhoid and other gastrointestinal tract infection. So the location, here is the location of the study sites. So this is the Batam. Batam is near Singapore. Maybe you know the Batam quite well, friends from Singapore. And then Banjarmasin is this location of the Banjarmasin, and Ambon in eastern Indonesia. So we located in Jakarta. So we travel quite many times during the study. We visited our friends in Batam here in Elizabeth Hospital Batam. Visited our collaborators, conducting socialization, establishment of study, and sample collection. We also visited the colleagues in Ansari Saleh Hospital in Banjarmasin, as well as in Ambon with the friends from the Haulusi Hospital there. And this is the algorithm when we conducted the patient recruitment. So basically, all of the dengue-like illness patients in admitting to hospital in Batam, Banjarmasin, and Ambon were recruited, and we got this number. And then the blood samples were collected, and we tested the patient using the dengue NS1, IgG, and IgM rapid tests in the hospitals to screen them for dengue. Then for the positive samples, we projected to dengue virus serotyping by RT-PCR, and continued by dengue virus genotyping by sequencing of the whole genome or the e-protein genes of the viruses. While for the negative samples, we screens for other arboviruses by RT-PCR as well. And this we use the pan-alpha virus and pan-flavivirus. Why we use these two? Because in Indonesia, there are other arboviruses circulating. The alpha viruses, for example, the chikungunya virus, there are chikungunya virus, and also the flavivirus. We have two major flavivirus in Indonesia, that which is the Japanese encephalitis and also Zika, also West Nile virus. So that's why we also screen for other arboviruses, because those arboviruses basically has a similarity on the symptoms with dengue. We also collected demographic clinical hematology data from the patients and from the hospital. We conducted a questionnaire, using a standardized questionnaire, and also the meteorological data collected from the meteorological bureau in each of the cities. And this is the results. The first result is the dengue laboratory diagnosis. So first we group the patient based on the WHO classification for dengue diagnosis, the virologically confirmed dengue. When the patient got the NS1 and or positive for PCR, and then for probable dengue, we categorize the patient when they got the IgM and or IgG positive, plus the clinical signs of dengue, and non-dengue when the patient were negative for all. And this is the results. The first result also showing the difference between dengue diagnostics, confirmatory diagnosis in 3 cities here. So if we look at the dengue confirmed and dengue probable here, between the 3 cities, it's clearly different, because more dengue confirmed in Banjarmasin compared to the one in Batam, and also here in Ambon. So in total, we recruited 732 patients from 3 cities, and then dengue patients, which is confirmed dengue and probable dengue, accounted for 57.8, while the non-dengue were around 40%, 42%. So this message is that not all of the dengue patient, dengue-like illness patient were truly dengue. It can be infected by other diseases, non-dengue diseases. And other viruses detected during the screening was one chikungunya patient infection in Ambon. And we didn't get any Zika or Japanese encephalitis or other flaviviruses. Then to determine the temporal distribution of the dengue-like illness patients, so we analyze the data, the hospital data, the admission data, and plot them with the climatic data. Here is the rain average range in each cities, and we plot them. And here we can see that the cases, the dengue cases in Batam and in Banjarmasin, both of them are quite similar, in which they are mostly occurred in around September until May each year. So around by the end of the year until the first 5 or 6 months of the years. However, in Ambon is different. So this is Ambon. We can see here the peak is not at the end of the year, but we can see here the peak is around the mid-year until the end of the year. But all of this area, the dengue, still following the rainy seasons in each of the cities. So this data is, I think, we think that this is the first to recognized that Ambon has a different seasonal periods of dengue. And then in terms of patient age, this is a dengue confirmed patients, dengue patient in 3 cities. This figures also shows that it is different. Yeah, Batam and Banjarmasin, they are quite the same, affecting young adolescents here. Yeah, still with the median age of 16 years in Banjarmasin, and median of 17 in Batam, while in Ambon, children under the 10 years old are still the main patients of dengue. Median age is 5 years. So this is different. So we cannot again saying that in Indonesia the dengue is a children disease or adult disease. It's different. It's unique, and in each of the cities, maybe in other cities also different. And I think they all can be also dynamics in the age distribution between cities in Indonesia. The next is the clinical symptoms. Here is also the clinical symptoms between cities. We recorded the clinical symptoms here, and they are also different. You can see here the headache, malaise, loss of appetite, and so on is reported more in Batam and in Ambon, while in Banjarmasin is quite different as well. So basically the profile of the symptomatic dengue are different between the 3 cities. And then severity of the disease. So we stratified the patients, and here the pie charts also clearly shown that they are different. In Batam, there are more mild dengue fever in Batam, and about the equal number of dengue and dengue hemorrhagic fevers there. And in Ambon, there are more, little bit more dengue hemorrhagic fever. We didn't see any dengue shock syndrome there or any fatalities in all of the cities. And the serostatus here, so it's quite different as well. Batam, maybe it's reflected in the serostatus, more primary infection in Batam, and more mild dengue there. But in Banjarmasin, we found that more patients are secondary infection, and Ambon it's more in primary infection, although there is a bit more severe disease there. And we also assess the RDT. So RDT, we use RDT here, and NS1, IgG, and IgM, and I'll try to correlate and compare between the cities here. We'll only focus on this NS1 here, and the result is quite different as well. So in Ambon, it's quite low sensitivity of the NS1 here compared to Banjarmasin and Batam. So this is only, so it's also clear results here. And then serotypes, of course, this is quite clear. Here, the serotypes is data when we collected all of the samples and then did the PCR serotyping on all of the samples, and this is the results here. It's quite clear, different serotype distribution in those 3 cities. So in Batam, we got dengue 1 and dengue 2 in an equal number predominance. In Banjarmasin, dengue 3 is the predominant one, but in Ambon, dengue 1 is the predominant serotypes. So there are distinct distribution of virus in 3 different regions in Indonesia at the same times using the same method. So this is the clear-cut results that demonstrate the difference in virological aspects of dengue in Indonesia. So although there is a predominance of the serotypes, but we also revealed some fluctuation of dengue serotypes, especially in Batam. In 2018, we saw a lot of dengue 2 in Batam, and then by the end of the year and then moved to 2019, we saw many dengue 4 circulation in Batam. And then we correlate the symptoms with the serotypes here. So here the symptoms and the serotypes. This is the number of the samples with each of the serotypes. But in general, we didn't see any significant correlation between the symptoms and the infecting serotypes. And then move to the genetic of the virus. So to determine the genotype of the dengue virus, so there are many genotypes of the virus actually. So and the genetic relationship among the isolates, we sequence the whole genome and the gene sequence on 70 dengue virus isolates from each city. This is the breakdown of the samples. Yeah. So in total 70 samples here. So and this is the results. This is for dengue 1. Dengue 1 in Batam, Banjarmasin, and Ambon, basically they all genotype as genotype 1. So this is genotype 1. There are other genotype 4. Yeah. So these are other Indonesian isolates and the black color. But in this here, the Ambon and Banjarmasin and Batam with the blue and red and yellow colors, green color. It's clearly shown here that they are different virus. Ambon is clustered with Ambon isolates, and Banjarmasin is also clustered by themselves, as well as the Batam virus here. So there are 3 main clusters of viruses, dengue 1 viruses in the 3 cities. They are different viruses. And then also in dengue 2, dengue 2, all of them are the cosmopolitan genotypes. This is quite common in Indonesia and the main genotype in Indonesia. So again, there are also separation of the genotypes both in Banjarmasin and Batam. And Ambon still, yes, it's quite different as well from the one in Banjarmasin and Batam. So they are also different viruses in the cities. And then the last one is the dengue 3. So dengue 3 again, we can see the clear separation between Banjarmasin strain here, down here, quite a large number of strain with a similar genetic characteristics. And Batam samples, although there are some Batam samples closely related to Banjarmasin, but again, this is another Batam samples separated from other strains. So I think I can conclude this with the conclusion like this. This is basically the dengue epidemiology in Indonesia is distinct and unique across region, even during the same period. So this is quite important that within the same period, they are different. So in our study, among this number of sample, the patients, so 60% of them, about 60% of them were dengue, but the rest are unknown. So this is homework for us to know exactly what the etiology of the unknown patients. And then the clinical, of course, immunological, demographic, and diagnostic aspect are all different between cities. Yeah, and the dengue serotype distribution is also distinct. It's quite clear. And then cannot be generalized for the whole country. So we cannot say that Indonesia is now dengue 3 predominance. No, we cannot say that, but we have to look at where and when the outbreak occurs. So we think that the diverse geographic, climatic, demographic, and the quality of healthcare may contribute to the varied clinical, immunological, and virological features. And we think that the continuous disease surveillance, dengue surveillance, is important to fully understand the complex dengue epidemiology in Indonesia. Then I would like to thank our contributors, and this is our contributors, our team in Jakarta. They all are dengue and emerging virus research unit teams in Jakarta. We also would like to thank our collaborators from Cambridge University, then our collaborators in the hospital, Dr. Sotyaningsih, and in Batam, Dr. Yuan Warli, and Banjarmasin, Dr. Anna, and Dr. Ingrid in Ambon. And this study is funded by the DP, the Indonesia Science Fund, with the LPDP and also Astra. Okay, thank you very much for your attention, and I can end this share the screen. Thank you.